Good morning, everybody. You can see from the little handout that I also spoke about newborn screening in San Antonio and Houston. So usually I say I get to talk about my two favorite subjects, genomes and babies, but today I'm going to try to just focus on genomes. So let's get started. Let's see. So these are our objectives for today. Hopefully at the end of this you'll be able to define the genome and the exome. You'll begin to talk about indications for when to do whole genome or whole exome sequencing, and then understand a little bit about the current challenges that we have when we look at a genome or an exome and don't quite know what to do with the results. So at first I'm going to set the stage. So why are we even talking about whole genomes and whole exomes? And then I'm going to talk a little bit about translational research. That's where we take the findings from the laboratory and begin to apply them to routine clinical care. Then we talk a little bit about best practices. So what are the leaders in the field doing today? What are they doing that they're not quite sure about? And what do we want them to do in the future? So what kinds of research projects should we be proposing to our academic partners and clinicians who do research? And then we'll spend a little bit of time on what we call the known unknown. So we know there are about 100 million variants across the 6 billion base pairs that we each carry. 3 billion from our mom, 3 billion from our dad. We don't know what those 100 million mean. So that's part of the known unknown. We really have to think about genetics in a new way where we're not quite sure what these variants mean and what does that mean for what you tell patients and their families. And then we're going to talk a little bit about some special considerations. So to set the stage, we're going to just remind you what human variation is, that the human genome is actually mapped, talk a little bit about the technology advancements that got us here, and then give you some definitions of genomic medicine. We talk about genomic medicine a lot. The sort of news anchors on CNN talk about genomic medicine a lot, so we want to give you a few definitions and really define the scope of what we mean when we talk about that. So just to begin at the basics, genetics is a study of single genes and their effects. That's what most of us learned in high school and undergraduate and our graduate or medical education. But genomics is really the study of all the functions of the genes and all the other parts of the genome. You know, we used to think that only 3% of the genome was coding and the rest was junk DNA. Now we know that that junk DNA is actually filled with molecular signatures that turn on and off genes at critical times in development and at critical times across our lifespan. Genomics not only takes into account the sort of linear base pairs of ACGT, but also what's going on in the environment. We're all born with a baseline genome, but then when you grow up in an inner city, you may have changes to your genome. If you work on a farm and are exposed to pesticides or insecticides, you might have a change to the genome. So we have to think about the genome as sort of a living thing across your lifespan. And when we think about genomic sequencing, when did you get your genome sequenced? Did you get it in a few hours after birth? Did you get it when you were 18? Did you get it when you were in the middle of your life? So we really have to think about genomic sequencing in the context of a lifespan. So just some basic concepts of inheritance after Dr. Rose's elegant talk. This is sort of like a really rudimentary slide, but just to remind us that 3 billion base pairs from our mom and our dad. As geneticists, we think about autosomal dominant, autosomal recessive, sex-linked disorders, all of the sort of genetic things that you think about. We think about these in the context of the genome as well. As we think about often when we find things we don't know, we begin to look at the parents and the extended family. So that's where pedigrees are sort of coming back into use in genomic medicine, not only looking at the patient or the proband, but also looking at the family and seeing what those tell us. As Dr. Friedenberg said, I was a member of the Human Genome Project, so my kids were little, and you know, I always had around the house the sort of traces because we were working 24/7, trying to beat every other lab to get our chromosomes done first. So I always focused on 4 letters, ACGT, and I had twins, and they came home from preschool, and they were so excited to tell me, "Mom, there's more than just 4 letters." And they were so excited, and I said, I know, there's U, right? So ACGT, the T is replaced with a U when it's RNA. So, but I think they were talking about the whole alphabet. So, but just a reminder, every living thing has a genome. The genomes are different sizes as you look across different species, even plants, even viruses. So you can look across, the first line just tells you the species or what I'm thinking about. The second line shows you the number of base pairs, and then the third line shows you the number of genes. You can see we used to get a cover when we sequenced a new species. This is sort of old news. If you sequence a honeybee or another sort of target of agricultural interest, you don't get a cover anymore. You maybe don't even get it published. So you'll have to find new species and new genomes on the internet. There's also a cancer genome. So as we said, your genome changes throughout your lifespan. So when we begin to look at things like breast cancer, lung cancer, and look at the genome sequence of those tumors, they're very different. They're not only different for the type of cancer, they're different across different patients. So you may take 10 breast cancer patients, and they may have totally different mutations that are causing the breast cancer or related to it. So the human genome has been mapped. Those are my 4 favorite letters. I was part of the government team at Lawrence Livermore and Berkeley Labs in California. And so we sequenced about 5 individuals. On the right was the report from Craig Venter's lab, who was a commercial lab, who raced with us. And they actually sequenced Dr. Venter's DNA. And it's interesting that when they sequenced the first dogs, his dog was the first dog as well. So we were hoping he had more pets so we could keep that going on the commercial side, but I think he just had a dog. But just a reminder that when we think about DNA and how we go from single base pairs to all the way down to proteins, this is sort of the central dogma that I know you're all very familiar with, from a gene through transcription, translation, and then ultimately a protein that really forms the building blocks and the functions of physiology. So one of the things that I wanted to remind us is that there's this whole world of molecular regulation. So with 23,000 or so genes, when we started the Genome Project, we thought there were 100,000 genes or 200,000 genes, but we found there was only about 23,000 genes. How do we make the hundreds of thousand proteins? Well, we do that by doing different techniques, different alternative splicing. So we might have a single gene, but then we do different things to it through modification. So out of that single gene can come many genes. So we used to think one gene, one protein. Now we think one gene, many different types of proteins. So this is how we sort of do it. When we compare humans to rice, and I know you guys all picked this out, it was really quick, we have 3 billion base pairs, on one side of our genome from our mothers, and then rice only has 389 megabases, so a much smaller genome. But when you look at the number of genes, they have a much larger number of genes. So how does rice do it, and how do humans do it? So in humans, we use alternative splicing. So let me see if I know how to do this. So here's a gene, and then these are the exomes. Remember, exons. Remember when we talk about whole exome sequencing, we're just talking about sequencing those colored areas, not the areas in between. And what humans do is they take these exons and then they rearrange them. And this is called alternative splicing. So out of this one gene in this example comes 3 different proteins. What rice does is a different technique, gene duplication. So they actually start out, they actually start out with one gene and then they duplicate it. So instead of doing the sort of switching around the pieces of the gene, they actually duplicate that gene and then modify it a little bit after that. So I always thought that was really interesting that rice, has more coding regions, but we ultimately have more proteins. So what are we doing with this whole genome sequencing? We're really looking at human variation. You can easily see that all these individuals are different. Well, we're 99.9% the same. So when we look on the computer, we look at our DNA sequence, it's only 0.1% that's different. In that 0.1% lies the whole answer to diseases, to how we respond to drugs, to what we might be at risk for in the future, to what we might pass on to our children. And so all of that is related to human variation. I love all these pictures, especially the picture of the two athletes that was on the cover of Sports Illustrated when I was growing up. I always remember that picture. And then when I studied genetics, I thought about, they look very different, but they're very similar in base pairs. And they may have the similar risks for different types of diseases. So we can't just look at the phenotype. We have to look at the genotype. And you see the little girl on the right from Glee, as Dr. Rose talked about, this is Down syndrome or trisomy 21. You can see that she is an actress today. So we have a lot of great interventions, a lot of great ways to help out individuals who have that particular anomaly. This is also genetic variation. Just thought I'd throw that in there. No comment. So when I think about human variation after the Human Genome Project, as Dr. Friedenberg said, my job was sort of on the bioinformatics team. That's because they wouldn't let me in the laboratory. DNA today in the laboratory is done by robots, IT guys, bioengineers. So we as scientists kind of tell them what we want, and they do that and close the door. So I had to learn a lot about computational algorithms and how to do software design. And what we were looking for is really lining up DNA sequences and finding out where the DNA sequences are different. And this is what the Human Variation Project is about. So really there's no normal genome. Everybody carries multiple changes that have never been seen before. We think that with each individual there are about 60 novel differences. So when your significant other tells you you're special, you're really special. So you can tell them, I know, it's in my DNA. But these differences contribute to different risks. And as geneticists and as clinicians thinking about applying these sort of tests and results, we have to think about that. What are they placing my patient and that family at risk for? And how can I use this to help determine what kind of therapy or management treatment might be useful for this patient? Another item that we always think about in genetics is this actionable? So if we sequence a genome or an exome, we come up with 100 million answers. Which of those are we going to act on today? If it's a newborn, we might act on a few of them. If it's an 18-year-old, we might consider other genes. It really depends on the phenotype, the timing of the test, and the other things that are going on for that patient. So just a reminder, there are many different types of genetic variation. The most common type is single nucleotide polymorphisms, or SNPs. And I don't know if this is too light to see, but an example is sickle cell disease, where there's a single base pair change that happens to occur in a coding region, and it changes the protein. There's also insertion deletions. This is just an example of cystic fibrosis. The most common mutation in Caucasians is a deletion of 3 base pairs that deletes a whole amino acid. And so when we look at the variations as clinicians and bioinformatics experts, we can say, does the mutation fall in a coding region? Does it fall in one of those molecular signatures? So if I changed a base, would it make a difference? Or does it fall in a region that I'm not quite sure what the function of that part of the genome is? There's also structural changes, and this is just an example in chronic myelogenous leukemia. So you see there's no base pairs that have been changed. It's actually large segments of the chromosomes that have translocated and changed the actual gene product and the protein product. And then there's copy number variation. So these are large segments of the genome. I think this is why we couldn't quite get our chromosomes done. We didn't understand when we were doing the whole genome project that there were these large, variations. And some of the variations we don't know what they do. They don't cause any effect on the phenotype or anything that we're starting to measure. But these large segments, whether they're deleted or whether they're duplicated or even, 4 copies of 1,000 base pairs in a row, we're not quite sure what those do yet. But that's another area and another type of genetic variation that now we think about. So the goal of using all of these new technologies, whether you're looking at the whole genome or the whole exome, is to really think about all these different types of applications, from what we call single gene disorders all the way to more complicated disorders like asthma, hypertension, and diabetes, where we think that there's not only one, but maybe a handful or maybe hundreds of genes that actually interact in an individual patient to put them at risk for these diseases. We also want to use this type of technology for what we call pharmacogenomics, and this is choosing the right medicine for the right patient at the right time. They used to call it personalized medicine. So it's all about, can we choose a medicine based on your genetic profile, or can we dose the medicine based on your genomic profile? So pharmacogenomics is kind of one of the things that have risen to the top. There's actually some requirements to do genotyping in drug labels. So there's a drug for HIV therapy that requires DNA testing before you can prescribe that drug. Coumadin, or warfarin, now includes a black box warning and recommends that you do genetic testing to help you dose that actual drug, and you can get to a stable PT-INR a little bit quicker. So pharmacogenomics is one of the things that we use routinely in clinical genetics. Cancer is another application that has come along very far, so we understand more about how to apply targeted sequencing in familial cancers as well as in sporadic cancers. So we're able to do this because of the technology advancements. So in 2003, we were just looking at a handful of genomes, but now we have the technology to look at individual patient genomes and to begin to think about how can we improve health outcomes for these patients. I always say genetics or genomics is just another tool in your toolkit. It's not the answer. So we're not going to do the genome and get, aha, I know what's wrong with this patient, I know what to treat, they're going to do great. It's just one of another piece of knowledge that you as clinicians and healthcare practitioners sort of have to think about. But we think it's an exciting one. So we're able to do this because of the significant decrease in the cost to do the whole genome, as well as all of this equipment. So robots, microfluidics, everything on a really small scale, parallel sequencing. We're able to do this. When we did the human genome, we sort of broke up the genome into big pieces and just went one by one. And now they break it up into really small pieces using sound waves, and then they do parallel sequencing. So they might sequence one region of the genome 1,000 times, and now they can do that in miniature and in parallel. So the actual sequencing of the genome is really quick these days. So this was just a headline that says, soon it will cost less to sequence a genome than to flush a toilet, and that will change medicine forever. And another one, when your genome costs less than your iPhone, the beautiful, terrifying future of DNA sequencing. So I love all these titles. You can find many on the web, but we want to make sure that some of this comes true, but that we understand how to make it beautiful, but not terrifying. So as I use this slide that really shows how much the sequencing has come down, and there's been this race to get to the $1,000 genome, and we're about there. What we see on the clinical side, even though we can do it, this is the interpretation. So now we're giving 6 billion base pairs to Dr. Powell and asking her, what does this mean? She's not going to stay up all night and try to look through all those sequences. We now have a big bioinformatics challenge. So using the phenotype, can we hone in on a few of those 23,000 genes, or do we have to look at the whole genome and try to figure out what all these variants mean? So it's not as easy as we think, and that's why we are in data overload. So we're changing my 4 favorite letters into 2 numbers. So this is what our computer sort of counterparts do. We love to work with IT guys and with geneticists and bioinformatics, so they can take all of this great knowledge from the laboratory and then help us as clinicians begin to understand it. So bioinformatics is really the integration of computers, databases, and softwares. If we had the technology to sequence the genome, but we didn't have the technology on the computer side, we wouldn't be as far along as we are. We really need to have those computational algorithms to begin to understand what these sequences mean. Just a few key definitions before we go further. So whole exome, or WES, it's really the analysis interpretation of the DNA that codes for proteins. So you can see in this picture over here, they use sort of the analogy of an orange slice or an apple slice, and the whole genome is the whole orange, but if you just take a slice out, that represents the exome. We think the exome is about 1% of the genome, but some literature reports think that about 85% of the disease-causing variants reside in the exons. You might remember that number and in 10 years say, Dr. Brower didn't know what she was talking about, it's only like 2%, because then we'll understand what the rest of the genome does. But today, we think that most of the disease-causing variants reside in the protein coding part of the genome. And then the whole genome is really the analysis interpretation of all the DNA sequence, so the whole orange. This is just a graphic that shows us the different areas and the different type of information we get back from all these different techniques. So today we're talking about whole genome or whole exome, but there's also other ways to target into the genome. If we sort of think about what the patient might have as a phenotype, we might be able to not only do whole exome, we might be able to just do targeted sequencing of a few candidate genes or targeted sequencing of a change that seems to happen in this particular family. So this just shows that there's a lot of different variation. I dropped my prize. That's okay. This just shows us that there's a lot of different information, and it's not just one test. I think that's what I was trying to get with this slide. This also shows us not just one test, it's also not just one technology. So this shows a comparison of 3 different platforms, Nimblegen, Agilent, and Illumina, and shows sort of a schematic in the A part of how they actually sequence the genome, and then it shows you for different annotations of the genome, how much of the genome they cover, because every technology doesn't cover the genome in the same way. So you have to think about when you get those results from your laboratory, think about what technology did they use, and is there anything you need to know in particular about that technology. So genomic medicine, what are we really talking about? We're talking about just really including genomic information into healthcare. Not everybody needs to be a geneticist to use genome sequencing information. Obviously, that's why many of you are here today. So we hope that the promise is that genomic medicine will help us do everything in the box and maybe even more. So it seems like a really big goal to detect disease earlier all the way down to reduce overall healthcare costs. I didn't write these. These are from a paper that talks about the definition of genomic medicine, but that's sort of what we think about. How do we use genomic medicine as another piece of information to ultimately improve patients' health care? We also think about genomic testing across the lifespan, whether it's in the first few hours after birth all the way to the hours before death. So how do we use genomic sequencing to improve health across the lifespan? We also want to think about the technology. What's the diagnostic yield of a whole genome or a whole exome? What are groups doing to help us understand or improve the diagnostic yield? And then what types of efforts could we think about that really help us translate basic discoveries or our basic understanding of common diseases to clinical care using genomics? So first, just some definitions for diagnostic yield. We really think about 2 things, analytic validity and clinical validity. So analytical validity is our ability to reliably identify variants. Clinical validity is to reliably associate those variants with disease. Those are 2 of the hallmarks of any laboratory developing a molecular test. Do you know what you're testing for, and is what you're testing for related to the disease that you're looking at? The sort of third rail of that is clinical utility. What's the cost and benefit of doing that? But I kept that third rail off of there. we'll think clinical utility is across the board. So this was a study that actually looked at how do we understand what these variants mean. This was a study that looked at 1,500 probands. These were people that actually had diagnosed diseases, all the way from cardiomyopathies to hearing loss to different disorders. And what they wanted to do was sequence the whole genome and see what kind of variants they found. Well, what they found in these patients who clinically had these conditions, they found that 68% of the pathogenic or likely pathogenic variants are only seen once. So really, you are special. And what's causing your disease may be special too, but that also increases the burden and the complexity when we're trying to analyze the data. It also showed in the study that 96% of variants are seen less than 10 times. So when you do whole genome sequencing, you're not going to see a huge answer that says, hey, look at me, out of 6 billion base pairs, I'm the one that is causing your patient trouble. You really have to think in the context of the correlation between the phenotype and the genotype, what kind of candidate genes, what kind of proteins, the disease affects in your patient. All those sort of basic rules of systems biology, you have to think back to what might be causing the issue. And then you can see in these pie graphs up here, it's sort of how we think about when we get the genome sequencing results. We think that in this study, 52% were benign, 17% were pathogenic, and 31% were variants of unknown significance. So these are the types of labels that you may get to sequencing that you get back from your laboratory in a result. So what are we trying to do to really annotate these variants so that we can tell clinicians and healthcare teams what we think as laboratorians might be important? So as we said, there's greater than 100 million variants. Most of them we don't understand, but at the federal level, there are two big initiatives. One is called the Clinical Genome Resource, and the other is called the Clinical Variome, or ClinVar. So these two efforts are really looking at taking these 100 million variants and starting to annotate them. What gene are they in? Have they been associated with a disease? Are they part of a new finding? Are they part of a drug target? So this is really creating databases that you can all access. So you can look up a particular mutation, a particular gene, and you can see all of the literature that's been reported about that particular sequence. It's also working with laboratories around the country. As you know, Baylor does a ton of whole genome sequencing. They're working with the professionals at Baylor to put every sequence variant they find into this database. And then they're having researchers say, have we seen this in the literature before? And they're attaching that literature to these variants. So we're slowly building this body of knowledge so that we can interpret the genome in a way that makes sense. It's really a combined resource, so taking all of the great minds and everything that we know as geneticists from OMIM that many people use to put in phenotypes and then try to come up with a diagnosis. So it's combining all the best of the best from the past into one particular database. So at ACMG, at the other projects that I do, we're all trying to help build this resource, and each one of you can do that on your own also. So as you get results back, you can go in, you can log in, you can put that patient's mutation in, what their symptoms were, and help us build that resource for the future. All of this comes down to translational research. So how do we begin to apply the discoveries to clinical care, and what are you going to do with those 100 million changes? So what ClinGen and ClinVar groups came up with is sort of this ranking of these variants from pathogenic all the way to benign. They do this through looking through the literature, through scientific reports, through does it change a protein product? Is it in an untranslated region? Is it in a promoter region? What does that variant actually do, or what is it predicted to do? This is just an example of a report on a whole exome sequencing. So as a clinician, this is actually what you might get back. You might get back a report that talks about there might be 2 disease-causing mutations that are related to this disorder, all the way down to there might be 300 to 600 variants in non-disease genes. So seems a little useful, but maybe still, a lot of homework to do. And I think this is where we're at today with whole genome and whole exome sequencing. We still have a lot to figure out on the interpretation side. So just some translational research targets. All of these are sort of diseases that you all work with every day as geneticists and clinicians, and trying to think about them in the context of, if we're now able to do whole genome or whole exome sequencing with these patients, what are the types of things we would want to do? So looking at the dystrophies, we know in the literature there are 4 main genes that have been reported. We know what some of the clinical correlates or phenotypes are, and then we know what research is trying to do. They're trying to develop drugs that actually skip the exons in kids with Duchenne muscular dystrophy. They're trying to work on gene therapy for some other types of dystrophy. So can we begin to use whole exome and whole genome sequencing to accelerate this research effort? And I'm just going to click through a few other examples. So with critical congenital heart disease, which is now part of routine newborn screening across the United States, we know that there are several genes involved. We know that there are several phenotypes or disease manifestations, and we know that what we're working on in the future, ultimately, earlier diagnosis and better treatment. So if we are able to apply these technologies, we'll be able to advance that quicker. So you can see that survival for kids with clinical or critical congenital heart disease is already improving because of the techniques used in the laboratory and by clinicians in surgery. But if we knew more about the genetic or genomic basis, will we even do a better job? Another example is diabetes with 4 key genes that have been reported. Many other genes are related to different types of diabetes. So if we knew more about the sequence of all the patients who have differences in their diabetes presentation, will we be able to design targeted therapies, or will we be able to design targeted management treatment styles for those patients? Another thing to look at is, your basic congenital anomalies. So thinking about all the genes that we know play a role in things like facial development, are there things that we can do in the future using whole genome or whole exome? Another is just an example is brain development. So this is a really big area, just a few of the top genes, thinking about these different phenotypes all the way from autism, epilepsy, schizophrenia, and thinking about how do we diagnose earlier, how do we do a precision diagnosis, how do we design new drugs and new therapies. So this is just an example with a seizure patient. So then turning to really looking at what are clinicians doing today? How are they using whole genome and whole exome? So just an example, as we think about genomic sequencing across the lifespan, we'll think of our first one in the first hours after birth. So Dr. Paul will tell you much more about genomic sequencing in the newborn period, but there's one group in Kansas City at Children's Mercy Hospital who's been doing rapid genome sequencing or rapid whole exome sequencing of babies in the NICU. And their idea was to diagnose those babies who have a genetic disorder much more quickly, so they could begin therapy and hopefully improve the outcomes of these babies. So this Kansas City group actually developed a bioinformatics approach that helps them get the result in 18 hours. They say it's the fastest genome sequencing and interpretation, those two things go together, in the world. So that's in Kansas City today. They're getting more visitors to that laboratory to try to figure out how they're doing it. And one of the ways they're able to do that is they take the phenotype. They're not looking at the whole genome. If they know what the phenotype of that baby is in the NICU, they're really honing in on some genes that they think will be of interest. But this was named in, I think, 2009 as one of the top 10 medical advancements by Time magazine. So this is really a group that has sort of figured out how to apply whole genome in the NICU. So thinking about how do we use it for diagnosis. So today, as a clinician, when you see a patient with developmental delay, there's sort of a tiered approach that you do when you analyze or try to figure out what's going on with that individual case, but through all the metabolic testing, through all the targeted testing, you ultimately, for some, don't come up with a diagnosis. If you're able to do whole genome or whole exome, we think that you're able to improve the diagnosis for 15 to 25% of kids. So that's what the reports have told us. So if we're able to apply this technology for kids with developmental delay, we may get to the answer for why they're delayed, and that may help us design therapies and interventions. When we think about treatment choice and whole exome or whole genome, one of the key areas is cancer. So this is just a schematic that shows you the development of cancer. Year 0, healthy tissue, healthy cell. Year 3, the cancer process has begun. By year 6, it's actually large enough to either feel or visualize through a screening test. And then year 10, it becomes a malignant tumor with metastases. So you can see, when I give lectures to high school students, I say, someday you're going to think we were strange, that we palpated to feel tumors. You're going to be able to see those much earlier, or see the predisposition to tumors much earlier. But what this shows is we really have an intervention window when the cancer or the tumor is much smaller. So when we look at these Kaplan-Meier curves and we begin to look at the genome and we begin to compare what kind of therapy did we apply for this patient, with what type of genetic change are we seeing in their cancer tissue. And this sorted out this group of patients. So this group of patients had a particular type of cancer. Some of the tumor cells in the patients had lost the short arm of chromosome 1. Some of them, patients, the short arm of chromosome 1 was still there. So they had the same type of cancer, but the molecular signature of the change in the cancer was different. And what we see is a different survival rate for one drug. So this helps us more quickly. For these people in the red, we want to do a different therapy. We don't want them going 5 years on a therapy that we now think is ineffective. We want to know this type of answer a little bit sooner. So it's really looking at the genome of a cancer and trying to target the therapy. So this is a pretty dramatic picture that shows a before and after treatment. It's a heat map that shows, tumor cells are very cellularly active. So it shows the heat map, and then after a few treatments of a targeted cancer drug that specifically targets the mutation in that cancer, you can see that the tumors have really shrunk. So that's, some of the goal in cancer therapies. There's a lot of federal initiatives going on, and these are just a few that I thought I would mention. One is the precision medicine project that President Obama announced in March. This is a $215 million investment to really combine the sequencing of the genome with the technology on the computers, and then to build tools for interpreting these large data sets. So today, just an internet click away, there are thousands of people's whose genome sequence you can mine through. You may put your own genome up there someday. You may get your genome sequence and say, hey, I want to be part of this national database. I want to advance understanding. I want to put my phenotypes in there. I want to help advance the knowledge. But today there's about 10,000 sequences from different people around the world in a database. The goal of this effort is to begin to sequence many, many, many more individuals and to look at their electronic medical records and see what kind of diseases they have. So can we sequence, 10,000 people with asthma, 10,000 people with diabetes, and begin to put all that information together and understand more about those individual diseases. The CDC also has a lot of efforts. So they're focused in newborn screening. So they actually have some grants out right now to public health labs to understand, can you do genomic sequencing in the newborn period in a state public public health lab, and what does it add to the knowledge that you get from routine newborn screening. They're also just announced an initiative to begin to sequence new viruses and to understand more about the changes in the virus and how that impacts both vaccinations and treatment for those different viruses. So there's a lot of initiatives across the federal government to begin to help us not only sequence more things, but to understand what the sequences mean. So now we get into sort of the known unknown, and this is the parts of the genome that we're not quite sure what to do with yet, and also the parts of the genome that we know very well what to do with. So ACMG had a work group that focused on the incidental findings in the clinical exome and genome sequencing. And what they came out with was this policy statement a little bit earlier this year, and they have 56 mutations that they say if you're sequencing a patient, and it might not be for any of the indications that these 56 are tied to, you should report out those 56. So included in there are many different types of genes. You can, search for this on the web and look through the whole table of the 56 different types of diseases. But you can see at the top BRCA1 and BRCA2. So that brings up some ethical considerations, right? If you're testing for something, a genetic mutation or a phenotype not related to breast cancer, but you find BRCA1 or BRCA2, ACMG says you should report that back. So that's a challenge on the counseling side. It also has implications for the family. It has implications if you do genomic sequencing in the newborn period. Should the parents have that knowledge about the infant before they're able to consent? What would the parent do? When would they tell them? There's a lot of different things that go into that. But this is sort of, a marker that ACMG came up with. That list of 56 may grow. It may shrink. It's going to change. We know that for sure. They also had, in 2000, sort of a nice paper that helped laboratories think through what do we do when we find variants that we weren't expecting or that we don't know what to do with. So this tells the laboratories and the clinicians how to think about variants that we're not quite sure of. And what they tell us to do for what we call variants of uncertain significance is to evaluate them in the context of the family history. So back to the pedigree, back to thinking about what is this patient actually experiencing, what am I trying to do with this type of test. They recommend testing other family members, so the routine thing you do in genetics, reviewing the literature relative to this gene function, and to reinterpret in the future. And so you can see for these, that's where the ClinGen and ClinVar project is so important. Hopefully, when we get a database that's shared, that's accessible, that has a lot of different knowledge pieces in it, we're going to be able to shrink the number of variants of uncertain significance and give more information back to the patient and the family. There's a lot of web-based resources that I wanted to remind you all about. There are several institutes at, the nation's research institution, the NIH. One of the institutes is especially focused on the human genome, and it's the National Human Genome Research Institute. They have 9 branches. Each of these branches has a different website, a different web page, different databases, different tutorials. If you want to learn more about particular areas of genomics, you can go to this website. It has some really great information. There's also some resources for genetics professionals. So all the way from, what you should know as a health professional, whether you're a geneticist or a physical therapist or a nurse, it sort of gives you the baseline of what we think you should know about genetics in it to being able to use genetics and genomics in your routine care, all the way through cancer resources, all the way down to what most of us are familiar with the online Mendelian's inheritance in man. That effort is also being brought into the ClinGen effort. So all of these are being linked through that centralized database of variants. So there's a lot of special considerations. This isn't an exhaustive list. I know you guys can probably come up with even more than this, but we talked a little bit about informed consent. So if you do a minor, whole exome, whole genome, did they consent to do that? If you do a whole genome on somebody who doesn't quite understand DNA sequencing, or that you might find something that you're not looking for, you might find non-paternity, you might find, other things that implicate the family for different types of risks for diseases. So informed consent is a huge area. Many of these genome sequencing teams are now ethicists, attorneys, psychologists, really helping us think through, as we bring this technology to families and to patients, what are the ethical considerations and what do we need to plan for? How do we really safeguard that knowledge, make sure the patient understands what they're consenting for, and make sure they understand the implications for themselves and for their families? There's also issues of data handling. So those IT guys building those databases, if I have my genome sequence done when I'm 18, what if I want to look at it when I'm 45 or when I'm 72? Who's maintaining that DNA sequence? Do I have it on chip, on a card? Do I put it in the cloud? What do I do? So there's a lot of groups trying to think through, how do we make sure that data is accessible for reinterpretation or for analysis as the patient goes through their lifespan? And then we think about return of results, as we just talked about, with the either 56 genes that we should return results on, or the variants of uncertain significance. So we need to think about, what's the context? What do we tell patients? We may find 50 million variants. How do we even tell them that? What do we start out with? How do we begin that conversation? And then how do we think about adult onset disorders? So if we sequence in the first hours after birth, or sequence some 18-month-old or 3-year-old with developmental delay, what if we find genes that are implicated in disorders that have an adult onset? What's our duty as clinicians, as geneticists, to tell the family, to help them understand that in the context of that baby's life? And are we going to have, it goes back to sort of what we thought about years ago, the vulnerable child syndrome, is if the family knows that they have an adult onset disorder, are they going to treat the baby differently? Are they not going to let him go out for sports? All these different implications on sort of the psychological and child development side. And there's many other considerations I'm sure that you guys will think of as the day goes on. For the genetic counselors and the physicians and the nurses and the, all the other allied professionals, there's a lot of counseling challenges. What we hear today from people who are actually consenting patients is that to consent takes a long time. And as Dr. Rose says, she's often has a few minutes with patients. So how do you cover everything you're going to cover, and how do you make sure they know what they're consenting for? Another challenge is result interpretation, making sure we understand that genomic sequencing has a limit. We may say we know the 6 billion base pairs of the genome, but we don't know. The ends of the chromosomes, the middle of the chromosomes are very hard to sequence. We're not yet finished with the genome, even though we said we were finished. We're still learning more about how to get through those tougher areas of sequencing. But also, there's a limitation of any laboratory test. There's false negatives, there's false positives. How do we put those in a context that patients can understand? How do we help them understand unanticipated results, whether it's a non-paternity, an adult onset disorder, or something that, causes them to have to make a decision right away about the care of that individual or the care of themselves? And then how do we think about variants of unknown significance? So that is always going to be a moving target for us on the side when we implement these types of technologies. So hopefully today we talked a lot about genomes and exomes, all the way from sort of, we have the technology, check. We have bench to bedside, maybe, sort of the approach figured out. We have a lot of federal funding behind it. We're trying to figure out, how do we make sure that everything we find out is ready for prime time? We don't want to implement something in the clinic that's not really proven. We don't want to do research on our patients. We have some guidelines, but we don't have enough guidelines. It's always a moving target. As we learn more, as the knowledge base grows, we have to have professional groups come out with guidelines, come out with best practices, tell us as a group what should we do with this type of technology? And then there's this whole area of ethics. How do we think about safeguarding the rights and the autonomy and, the health outcomes and sort of choice that these patients and their families might have? We're learning a lot about that as these technologies get implemented, but we're going to learn a lot more. And I'm glad that many of the efforts that are funded are actually including this as one of the main focuses. And then we have a wish list of future efforts. So we might want to do whole genome sequencing in many different types of disorders. We might want to do it for many different types of drug development. How do we make sure that we have the technology available and can harness the understandings from that? We're not in the mode anymore of sort of doing a whole genome, publishing, 10 years, do another. It's all coming very fast. And we may see most of the whole genome and whole exome sequencing being done in the clinic, not being done by researchers. So we want to provide a way for clinicians to put those variants in that central database so we can begin to understand the types of things that they're finding. So I'm here representing the American College of Medical Genetics and Genomics. You can check us out. We have a lot of tutorials online about genetics and genomics, and you can email us if you have any additional questions. A lot of the work that I do is funded by the Eunice Kennedy Shriver National Institute of Child Health and Human Development, where we're focused on pediatric research and research related to newborn screening. And when I was in graduate school, Dr. Sanger was on my graduate committee. We lost him this year, so I just wanted to include this just as sort of a tribute to him. He was one of the founders of a technology called FISH, or fluorescent in situ hybridization, and that's where we could visualize the chromosomes. We could look for changes at a visual level, look for changes that are hallmarks for different types of cancer and disorders, and this was actually his license plate, O to be fishing. So both he liked to fish and he liked to do fluorescent painting of chromosomes. So thank you for your time today, and thanks to the Texas Department of State Health Services, the Texas Genetics Program, Dr. Friedenberg, and our colleagues at the Texas Newborn Screening Division. Thank you.
Thank you, Dr. Brower. Questions?
So my question was about the whole exome sequencing, because I think when we interpret like the genes screening or testing, you tell us there's a mutation, so we know what to do. So and you showed us a report on the whole exome sequencing. So is there any information of a direct correlation between with your exome sequencing results and any clinical diseases, or for clinicians is it still too early to link them to diseases?
There's a lot of information about whole exome and individual diseases, and you can go to the ClinGen and ClinVar resources and find those. So you can actually search for a mutation, tie it to a gene, tie it to a disorder. So we're building that knowledge base, but there's a lot of information for, we think there's 4,000 genetic conditions. There's information tied to each one of those in that database. Some of them have much more information than others, and some of them have new mutations. We're always discovering new mutations for disorders we thought we understood, but it's a great resource. So I would encourage you to check it out.
So when you use the word mutation, I guess generally we think of as a gene mutation. So I guess the new terminology is an exon or exome mutation as well as something new we need to think about, I guess.
Yeah, and they recommend in that statement from ACMG that we don't say mutation anymore, we say variation. And so it's a habit to say mutation. Sometimes we think a mutation means it actually does something, right, in the clinic, but we think now about a variant and what does that variant do to the DNA sequence and the protein, and in this particular patient, what role does it play in their disease progression?
Okay,
so,
thank you.
Yeah. Hey.
Something that actually I never thought about, that you mentioned the central dogma, and we really don't look at the replication cycle too much in the central dogma. Now, you mentioned that maybe we have to sequence our genome, or part of the genome targeted sequencing, several times during our lifetime.
Yeah.
Let's say that someone newborn screened with a variant. But later, those variants probably will change because of the replication cycle. What is the consequence of that? We will carry variants dynamically through our lifespan.
Right,
This means that we cannot really correlate anything to anything.
right. Dr. Rose, go back to, so I confused you too. Yeah. So I think it's true. I think one thing, on sort of my wish list is to see a longitudinal approach to genomic sequencing. Take the individuals or newborns that Dr. Powell is sequencing and sequence them again throughout their lifespan and begin to compare those and see what did it mean for that individual's life and the conditions that they have. So I think we don't know yet. So you bring up a really good topic. You guys are great, smart, thinking, way ahead of us. Thank you.
So
Oh, good.
here's Dr. Brower's prize.
My prize.
Thank you, Gift, for being with